Okay, welcome everybody to Chapter 12 from Gunstream, where we're going to discuss the anatomy and physiology of the circulatory system. Specifically, we'll be dealing with the heart and the blood vessels. We'll ID the protective coverings of the heart and the parts of the heart and describe what they do. We'll describe the events that take place during the cardiac cycle and trace the flow of blood through the heart. And we'll talk about the cardiac conduction system, which is what the heart uses in order to synchronize when and where it pumps. We'll explain how the heart rate is regulated. We'll talk about the different types of blood vessels: arteries, arterioles, capillaries, venules, and veins, their physical characteristics, and also their location in the body and the direction of blood flow in these vessels as well. And we'll describe how materials are exchanged between capillaries and interstitial fluid. We'll describe the mechanism of blood circulation. We'll compare systolic and diastolic blood pressure, describe how blood pressure is regulated, and we'll talk about some of the different pathways that blood takes through the body. So we'll be looking at some of the major arteries and veins in the systemic and in the pulmonary circuits. We'll ID the major systemic arteries and the organs and body regions that they supply. We'll ID the major systemic veins and the organs and body regions they drain, and we'll talk about heart disorders to wrap up. So, what is your heart at its core? Okay, it is a blood pump. We have a closed circulatory system, which means that the blood that's pumped by the heart is always contained in some kind of a chamber. The chamber could be the blood pump itself, it could be an artery, it could be a vein, it could be an arteriole, a venule, or it could be a capillary. So, in order for blood to serve its purpose as a delivery vehicle for nutrients and oxygen, and a removal mechanism for waste products such as carbon dioxide, urea, and other metabolic wastes, what has to happen is that at the level of the tissues, we have to have the capability of unloading components from the blood into the interstitial fluid and retrieving waste products from the interstitial fluid and loading that into the blood. And we'll see that in the human body that happens primarily where the capillaries meet up with the systemic tissues. Not all organisms have a closed circulatory system. Some organisms have an open circulatory system where the blood literally bathes all the different organs. Insects, for instance, have this type of circulatory system. But we have a closed system. The heart and the blood vessels are part of it. The heart pumps the blood, and the arteries carry the blood away from the heart and to the capillaries, while the veins bring the blood back to the heart from the capillaries. Okay, so remember, always, 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 when we're talking about arteries and veins, there's always a difference in terms of direction, right? Veins always bring blood back to the heart. Arteries always carry blood away from the heart. Don't get caught up, as many anatomy and physiology students do, with the color of the blood that's in those vessels, okay, because that can sometimes trip you up. We're only concerned primarily with the direction of flow, okay. The heart has 4 chambers. It is a muscular pump. It's made up of cardiac muscle tissue, which is striated but under involuntary control. The heart has the ability to create its own cycle because it has embedded in its walls tissues that are electrically active that synchronize when the 4 chambers of the heart squeeze. But independently of that, the autonomic nervous system, which, as we learned in the previous chapter, is responsible for the involuntary maintenance of homeostasis, regulates, among many other things, the heart rate and the force with which the heart squeezes. And it does so by talking to these electrical components that are embedded in the walls of the heart. The heart lies between the lungs. It's superior to the diaphragm. The apex extends to the left side at the level of the 5th rib, and the base lies at the level of the 2nd rib and is attached to large blood vessels. So, in a sense, when you look at the heart in anatomical position, it's kind of upside down. You normally think of the base of something being at the bottom. In this case, the base of the heart is actually on the superior aspect. Remember from the early chapters on the body cavities, the lungs lie in the pleural cavities, the right lung in the right pleural cavity, and the left lung in the left pleural cavity. The heart lies within the mediastinum, which contains the pericardial chamber, okay? And that is essentially what surrounds the actual beating heart muscle. This entire chamber above the diaphragm is the thoracic cavity. Okay, so don't forget that. Okay, the heart has protective coverings that allow it to carry out its task of pumping blood without irritating and inflaming itself. And these protective coverings do a couple of things. They lubricate the heart as it beats so it doesn't irritate, inflame, and scar as it pumps the blood in a ringing motion throughout the body. And it also serves to stabilize the position of the heart in the chest wall. The pericardium surrounds the heart and the associated blood vessels. There's an outer fibrous pericardium that's hooked to the diaphragm, the sternum, and the vertebral column, and it's basically made up of connective tissue. And an inner parietal pericardium that lines the fibrous pericardium. It folds back to form the visceral pericardium, which is the outer heart wall. So you can see here the different layers, right? You can see, if we look at the heart, this is a coronal section through the heart. This tissue here is myocardium. This is actual heart muscle tissue. This layer is endocardium. And then as we move out towards the superficial aspect, you can see the visceral pericardium, and then there's going to be a fluid-filled layer on top of it, which is filled with pericardial fluid, and then the parietal pericardium, and then the fibrous pericardium, the most superficial layer that attaches it to the surrounding structures. The parietal and visceral pericardia are serous membranes. They make a slippery fluid. It's secreted into the pericardial cavity that reduces the friction that the heart experiences every time that it beats. If we didn't do this, every time the heart beats, it doesn't just squeeze, it kind of twists on its axis, and it would rub against the surrounding tissue, and that would cause the muscle to scar and inflame, and eventually this would impede its function. So this reduces friction during contraction. The heart wall is made up of the myocardium, which is cardiac muscle tissue. That's what actually produces the force of contraction. The epicardium, which is that layer that lies on top of the myocardium, which is sometimes also called the visceral pericardium. So two names for the same thing. It's the outermost layer, and it contains blood vessels that nourish the heart. And then the endocardium, which is the inner layer of the heart chambers, which is made up of simple squamous epithelium. This also lines the insides of the blood vessels that are attached to the heart. Endocardium is special because it has the ability to repel the formed elements of blood so that they don't snag and rupture and release their contents, just as the linings of the blood vessels do. So let's look at the heart chambers. There are 4 total heart chambers. The 2 atria are the topmost chambers, and they receive blood from veins, and the 2 ventricles are the lowermost chambers, and they pump blood to the arteries. The ventricles are sometimes known as the discharging chambers because they are the ones that actually send blood out of the heart, out of the blood pump. And they do the majority of the work in the heart, and that's why they're stronger. You'll notice, for instance, that the walls of the ventricles, you can see here and here, are thicker than the walls of the atria. The ventricles have a couple of things working against them. Number one, they have to work against gravity in order to pump blood around the body. And number two, they have to overcome the back pressure of the blood in the great vessels in order to eject it. Okay, so there's going to be some back pressure, and there's also going to be the fact that you're working against gravity. So as a result, these chambers must be stronger than the upper chambers. So the atria, by comparison to the ventricles, are rather weak. They are able to work with gravity, and they don't have to send the blood through nearly as great a distance. The atrial septum and the interventricular septum are what separate these two chambers from each other. Now, just from looking at this figure, you'll notice that the left ventricle, which is this chamber here, is the most powerful of the four chambers. You can tell that by the thickness of the wall. And the reason for this is that the left ventricle does more work than any of the other chambers because it has to pump blood to the entire body, whereas the right ventricle over here only has to pump blood to the lungs, which lie at a very, very close distance from the heart. So they don't have to provide nearly as much force in order to perform their task. The atria, of course, only have to push blood from the upper to the lower chambers, and so they're relatively weak. The heart is a double pump. The left atria and the left ventricle make up the left pump, while the right atria and ventricle make up the right pump. Very important to understand that the amount of blood ejected by the left and the right side of the heart under normal circumstances is always the same. Okay? The right atrium isn't as powerful, but it doesn't have to overcome as much force against its pumping action as the left ventricle does. The left ventricle is more powerful because it has to overcome greater resistance. But the volume of blood that the right and the left side of the heart pump out with each beat is always the same volume under normal circumstances. The differences in the thicknesses of the walls depend on the amount of work performed by the chamber, and we've already detailed why the left ventricle is the most powerful of the four chambers, because it has to pump blood to the entire body, while the right ventricle only has to pump blood to the lungs and back. So that's a reasonably easy task. Okay. Now what you're looking at here is the back of the heart from the outside. And you can see some very important features looking at the back of the heart. Notice again the apex and the base. Notice that there are several large blood vessels coming out of the top of the heart. This big sort of U-shaped vessel is called the aorta. This is the largest artery in the body, and it serves as the pathway for blood to take to the rest of the body. And then you've got the superior and inferior vena cava. They bring blood back to the heart from areas above and below it. Okay, so these are two of the largest veins in the body. These are the pulmonary arteries. Now you'll notice that the pulmonary arteries are blue. And the reason for that is that the blood they contain has a relatively low oxygen content. They bring blood back from they take blood out from the heart and they deliver it to the lungs. Okay, so what's happening here is that the blood that's in these vessels leaves the right ventricle, enters the pulmonary trunk, which isn't visible from this side of the heart, and enters the right and left pulmonary arteries, where it eventually ends up at the lungs. Now what happens in the lungs is that the blood picks up oxygen from the air that you inhale, and that causes it to change color from a dark red to a very bright red, which indicates that the hemoglobin is saturated with oxygen. That blood then returns back to the heart through the left and the right pulmonary veins. You can see those pictured here, and they are going to empty blood into the left atria. Okay? From the left atria, the blood will descend down into the left ventricle, and the left ventricle will then eject that blood into the aorta. Okay? So the red color of these vessels indicates the oxygen content of the blood that they contain. Now, what are some other major features back here? Well, you've got the coronary sinus, which drains blood from the heart muscle itself and returns it to the right atrium. So that's a very important blood vessel. It's part of the coronary circulation. And then you can also see some of the major blood vessels that supply oxygen to the heart muscle. The left and right coronary arteries aren't visible on this side of the heart. They're on the front side, but they do wrap around to the back of the heart. The circumflex artery is one of those branches. You can see a portion of it here, and delivers blood to the heart muscle. So the heart, oddly enough, gets first dibs on the oxygenated blood that it pumps into the systemic circulation. And that's very important because the heart muscle is always working. So it needs a constant supply of oxygen and nutrients. These little flaps are called auricles. Okay, you can see the left auricle here. The right auricle is somewhat hidden. It would be in this general area. The auricles are ear-like flaps that cover the outside of each of the atria, and when the atria fill with blood, those auricles will expand, okay, causing the upper chambers of the heart to enlarge. Remember that the amount of blood that the atria receive is going to be equal to the amount of blood that is going to be pumped into the ventricles. Okay, so you have to have some area for expansion, because these chambers without these auricle flaps wouldn't be able to hold enough blood for the cardiac cycle to be efficient at pumping blood throughout the body. Okay. Blood flows in one direction through the heart due to the presence of one-way valves. The atrioventricular valves, as the name states, are between the atria and the ventricles. Remember, the atria are the upper chambers, the ventricles are the lower chambers. They allow flow from the atria to the ventricles, and they prohibit flow in the opposite direction. Now, it's very important to understand that the mechanism that allows this is the fact that these atrioventricular valves are stabilized at their inferior aspect by little structures called the chordae tendineae, which essentially hold the flaps of these atrioventricular valves, stabilize them so they can't open back into the atria. The chordae tendineae attach to muscles in the base of the ventricles called papillary muscles, and this is important because if these valves were leaky, then the pumping efficiency of the heart would be severely affected. And sometimes that does happen when the valves are not properly stabilized and permit backflow. We call that a prolapse. And we generally, sometimes we can leave the prolapse alone if it's not too severe, but if it's an extreme prolapse, we have to surgically intervene. We can either repair the valve or we can replace it. The tricuspid valve lies between the right atrium and the right ventricle, while the bicuspid is between the left atrium and ventricle. The names tri and bicuspid come from the number of flaps that each valve has, the tricuspid valve having 3 and the bicuspid or mitral valve having 2. Now you might well ask, what's this mitral valve jazz? Okay, why do we call the bicuspid valve the mitral valve? The reason we call it that is because the two valves on the bicuspid valve, when they're together, when they're closed, look a lot like a bishop's mitre. That's the ceremonial headdress that's worn by high officials in the Catholic Church. So that's where the name comes from. The semilunar valves are located at the base of blood vessels attached to the ventricles. The pulmonary semilunar valve is between the right ventricle and pulmonary trunk, while the aortic semilunar valve is between the left ventricle and the aorta. Okay, and again, the term lunar comes from moon-shaped. You can see here in the right-hand figure a view of the heart from the top with the atria removed, and you can see the semilunar valves. Those are here and here, and you can see the bi and the tricuspid valves here and here. Notice the 3 flaps on the tricuspid, the 2 on the bicuspid. And notice that they open in opposite directions. The aortic and the pulmonary semilunar valves only open out of the ventricles, so they only allow blood to leave the blood pump, the heart, while the bicuspid and tricuspid valves only open into the ventricles, so they permit flow from the upper to the lower chambers. There's a fibrous skeleton that serves as a sort of a framework for the heart. Chordae tendineae extend from the valve cusps to the papillary muscles in the ventricle walls, and that prevents the prolapse phenomena we were talking about earlier. The semilunar valves have three pocket-like cusps that prevent backflow when the ventricles relax. So you can see here how when these guys close, they're very unlikely to evert and permit blood to flow back into the ventricles because of their geometry. Okay, so it's not so much stabilization by connective tissue as it is with the bicuspid and the tricuspid valve here. It's the geometry of the valves that keeps them from prolapsing. Here you can see an anterior view of the heart, and while we're looking, we should probably trace the flow of blood through it while we're here. So let's choose the appropriate color. We'll choose blue for deoxygenated blood coming back to the heart. And what you can see here, inferior vena cava here, okay, deoxygenated blood flows in through the inferior vena cava and is going to enter into the right atrium, okay. Superior vena cava, blood's going to flow from areas above the heart and is going to enter the heart here, also into the right atrium. And we also can't forget that blood coming from the coronary sinus being drained from the heart muscle also comes into this chamber. Okay, where does the blood go next? Well, as the pressure builds inside the right atria, the tricuspid valve will open, okay, due to the difference in pressure, and the blood will flow from the right atrium into the right ventricle. The next thing that will happen is that the right ventricle will begin to exert pressure, and that'll cause the tricuspid valve to close and prevent backflow into the atria. And then once the pressure builds sufficiently, the blood will be ejected through the pulmonary semilunar valve into the pulmonary trunk, and from there we're going to head to the lungs. So let's write lungs over here on either side of the heart. So to the lungs. So there's blood going to the lungs one way, and there's blood going to the lungs the other way. Now, what happens in the lungs? Well, the blood is going to pick up oxygen from the air that you breathe. The air you breathe is about 20% oxygen, and it's going to change color from a dark red to a bright red. So let's change our color scheme here to red. That oxygenated blood is going to then come back from the lungs through the left and right pulmonary veins. There's a pair on each side of the heart. And will enter the left atrium. So oxygenated blood will come in here and fill this chamber, okay, from both sides. It's kind of hard to see here, but that's the left atrium filling up with blood coming from the left and the right pulmonary veins. Now, same thing's going to happen with the left atrium. As the pressure builds, the bicuspid valve will open and will permit blood to flow from the left atrium into the left ventricle, and then on its last trip out of the heart, the left ventricle will begin to exert pressure. The bicuspid valve will shut, and then the oxygenated blood will flow through the aortic semilunar valve, and then from here out to the body. Okay, all these blood vessels lead to various locations in the body. Okay, and we'll just put here to the body, B-O-D-Y. Okay, so that's the skinny on blood flow. It's kind of nice to see it with the heart opened up this way. And this continues throughout your life. Now, I've given the impression from the way I've described the beating of the heart that the pumping action goes right atrium, right ventricle, then left atrium, left ventricle. And that's actually not the way that the pumping action occurs. The two upper chambers of the heart squeeze at the same time, and then as the two lower chambers fill, they will squeeze at the same time, ejecting blood the right ventricle to the pulmonary trunk to the lungs, and the left ventricle through the aortic semilunar valve to the body. Okay, so it's always squeeze, 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 squeeze. Okay, but the path of blood is exactly as we've diagrammed it here. You can see the chordae tendineae that stabilize the cuspid flaps to the base of the heart. We sometimes call these the heartstrings. The chordae tendineae are pulled on by the papillary muscles, and that assists with the flow of blood from the upper to the lower chambers. And in addition to that, the atria, of course, give a little squeeze in order to eject the remaining fraction of blood that they contain after the blood spills from the upper to the lower chambers into the right and left ventricles, respectively. The cardiac cycle includes systole, which is the contraction phase of the ventricles, and diastole, which is the relaxation phase of the ventricles. Now you might say, wait a minute, what about the atria? Well, the atria undergo systole and diastole also, but we can't measure that at pulse points in the body. We can't pick that up, for instance, on the radial pulse or in the carotid pulse, because that squeezing action is too weak. So when we talk about systolic and diastolic pressure, and we talk about systole and diastole, we're generally referring to what the ventricles are doing. When the atria and ventricles are relaxed, blood flows into the atria through the open AV valves and into the ventricles. The semilunar valves are closed due to the pressure in the arteries on the ventricles that keeps them shut. And then what happens next is that atrial systole is going to eject the remaining fraction into the ventricles. Ventricular diastole, which is happening at the same time as atrial systole, increases the blood pressure in the ventricles, and it causes the AV valves to shut, and at the same time the pressure begins to build until the semilunar valves open. Now, there is a period of time during which both the AV valves and the semilunar valves are closed, and that period for the ventricles is called isovolumetric contraction, meaning that pressure is building in the lower chambers, but no change in volume is taking place because both doors into and out of the ventricles are closed. But eventually what happens is that the ventricles generate enough force to overcome that back pressure. The semilunar valves open up and they permit the blood flow out of the heart. Now, very important thing to point out is that in individuals that are out of shape, they have excess body weight, they may have deposits in their arteries that cause their arteries to stiffen and the lumens of the arteries to narrow, causes a lot of extra back pressure on the heart, so the heart has to work harder to pump blood out. And what can happen over time is that the heart muscle can actually enlarge. They call that hypertrophy. And the danger there is it could ultimately result in the heart failing. And so this is one of the reasons why we always recommend that you stay in shape, keep bad body weight in the form of fat off of you, and try to increase the percentage of lean muscle mass you have, because muscle actually causes more blood vessels to be laid down, and as a result that takes a lot of the back pressure off of the heart, allows it to pump the blood around the body with a minimum of effort. That's why when you take the pulse of a well-conditioned athlete, it tends to be low and slow, whereas an individual that's out of shape, their resting pulse is going to be relatively rapid. Ventricular diastole follows this. This allows the AV valves to open and the semilunar valves to close. Then the cycle repeats. There are some heart sounds that we should be familiar with as well. Lub-dub, and then there's a pause and another lub-dub. Lub indicates the closing of the AV valves during ventricular diastole. That's when the lower chambers are opening up, okay? And the AV valves close, okay, as the pressure builds inside the ventricles, forcing those valves shut. And then DUP is the closing of the semilunar valves during ventricular systole. Okay, so the heart is going to squeeze the blood out of the ventricles, and then what will happen at the end of that cycle is that the blood will actually rebound. It'll actually flow back towards the heart briefly, but because again of the geometry of the semilunar valves, they will close. You'll hear the blood bouncing off of those valve flaps, and that's the second heart sound. Okay, there's two basic circuits of blood flow. There's the pulmonary and the systemic circuit. The pulmonary circuit is where deoxygenated blood flows from the right ventricle to the lungs, and oxygenated blood flows from the lungs back to the left atrium. While in the systemic circuit, oxygenated blood flows from the left ventricle to the body, and deoxygenated blood flows from the body to the right atrium through the superior and inferior vena cava. So here we see the steps in heart circulation, just as we diagrammed them earlier, but this diagram has a little bit more detail to it. The superior and inferior vena cava drain blood from regions around the body and send it back to the heart. The superior vena cava draining regions above the heart, and the inferior vena cava draining regions below the heart. The right atrium begins to fill with blood. At the same time, the pulmonary veins return blood from the lungs to the right atrium, and then the atria will squeeze, pushing the blood into the ventricles. The right ventricle will pump the blood to the pulmonary trunk and thus to the lungs. The blood moves into the right and left pulmonary arteries, which notice in the diagram are blue, and this heads to the lungs. And the left ventricle at the same time is going to pump blood into the aorta, which carries blood out to the entire body. Notice that that blood is red because of its high oxygen content. So notice in this diagram, it's kind of nice, you can see what happens in the lungs to the blood. The hemoglobin is picking up oxygen from the air that you breathe, and at the same time it's releasing carbon dioxide, which is both bound to the hemoglobin and is also present in the blood plasma, and then we exhale that gas when we breathe out. So very important that this happen. The oxygen is critical because the cells need it in order to engage in aerobic respiration, which allows them to maximize the amount of ATP they produce per fuel molecule that they take in. Just to give you an idea of what the energy difference is, let's just do this in black. With oxygen, O2, we're going to generate between 36 and 38 ATP molecules per glucose that the cell takes in. Without oxygen, so without oxygen, you're going to make a whopping 2 ATP per glucose molecule. That's not enough to run the body. Okay? So if somebody asks you, why do we need to breathe? This is why. If you didn't breathe, you wouldn't make enough ATP for the cell to do any kind of useful work, and you'd die in very short order. The heart, remember, has first dibs on the blood that it pumps. The right and left coronary arteries branch off of the base of the aorta and supply the heart tissue, while the cardiac veins return blood to the coronary sinus, which dumps the blood back into the right atrium. You can see the coronary sinus here looking at this rear view of the heart. The right and left coronary arteries are only going to be visible in an anterior view, and we'll get one of those in a bit. Now, in order for the heart to be an effective pump, it needs to coordinate its pumping activity. The cells that make up the heart muscle have to squeeze in a coordinated fashion in order to send the blood from the atria to the ventricles and from the ventricles into the aorta and the pulmonary trunk. If we don't coordinate the action of the heart muscle cells that make up the myocardium, the heart would just essentially vibrate or shimmy. And there's a term for that. We call that fibrillation. And that's not an effective way to pump blood around the body. So the heart conduction system is specialized muscle tissue that acts like neural tissue in the heart. It's like the heart has its own pacemaker. They generate electrical impulses spontaneously that cause the myocardium to contract. And so what happens here is that the electrical components are the SA node, the AV node, the AV bundle, and the Purkinje fibers. And the way that this works is that the SA node normally serves as the heart's pacemaker, and it's going to generate a depolarizing signal periodically, about, let's say 75 times per minute on average. This depolarizing wave will run from the SA node across the walls of the atria until it pauses and is captured by the AV node. After this happens, after this electrical signal runs across the walls of the atria and pauses at the AV node, the atria will squeeze. Okay, so they'll engage in systole and eject their blood into the lower chambers. The AV node, once it's captured this electrical signal, will rocket it down the interventricular septum through the AV bundle and the right and left bundle branches to the apex of the heart, and then it'll radiate up the sides of the ventricles through the Purkinje fibers, where it'll talk to the papillary muscles and the musculature in the ventricular walls. And once that electrical signal moves through the walls of the ventricles back up to the atrioventricular septum, the ventricles will squeeze in unison and eject the blood, the right ventricle into the pulmonary trunk and the left ventricle into the aortic semilunar valve. The sinoatrial node is the heart's pacemaker. It forms the impulses that initiate the heartbeat that cause the contraction of the atria, while the AV node receives the impulse from the SA node, delays it in passing it to the node to allow the ventricles time to fill with blood, and then we send that electrical information to the AV bundle, the bundle branches, and the Purkinje fibers, and then up the lateral ventricular walls, and finally the impulse is relayed to the myocardia of the ventricles. The contraction occurs from the apex upward, and we eject the blood from the heart. Okay? So this would happen even if the heart had no nerve input whatsoever. Okay? This is within the walls of the heart. This is embedded tissue. But as you know, your heart rate changes depending on your activity level. The more active you are, the more rapidly your heart beats and the more powerfully your heart contracts. By the same token, the less active you are, the less rapidly your heart beats and the less powerfully it contracts. And that's to meet oxygen demands and fuel demands that the body puts on the heart. So how do we control this? Well, we control this by the influence of the autonomic nervous system on the SA and the AV nodes in the heart. The sympathetic division of the autonomic nervous system, which is nicknamed the fight or flight division, tends to accelerate the rate at which impulses are generated from the SA node, while the parasympathetic division tends to have the opposite effect. And so these kick in when our activity levels change or when our emotional state changes. Okay? And that's how it is we're able to regulate the heartbeat in order to meet our oxygen and fuel demands. An electrocardiogram is a recording of the electrical current generated during heart contraction, and it's performed by an electrocardiograph. The electrocardiogram has three distinct waves: the P wave, which represents atrial depolarization, the QRS wave, which represents ventricular depolarization, and the T wave, which represents ventricular repolarization. Now, what I have here in the inset is a nice little diagram that will show you exactly how the EKG is generated and what electrical and mechanical events are occurring as the heart beats. So if we watch, let's see if we can get this to go. Yes. Hold on. There's the P wave. This is the EKG down here. Okay, there's the QRS complex. And then this last wave is the T wave. Okay, so P, QRS, T wave. Now, the question becomes, okay, what else am I looking at here? I noticed there was stuff going on in the heart at the same time. Well, let's start it again, okay? And let's look at the heart this time. SA node fires, electrical impulse is captured by the AV node, the atria squeeze. The electrical signal goes down the apex through the bundle branches, and the Purkinje fibers bring it up through the walls of the ventricles, and then the ventricles squeeze, okay? So let's watch it again. One more time. AV node, or SA node, AV node, bundle, left and right bundle branches, Purkinje fibers, okay? And notice which waveform corresponds to which of these activities, okay? So the way to remember this, or the way I always taught myself to remember this, is to think of it like an old high school cheer. Depolarize, squeeze, repolarize, relax. Depolarize, squeeze, repolarize, relax. Depolarization represents the electrical signal that leads to the muscle squeezing, so it leads to systole. So we start in the atria, the walls of the atria depolarize, and then the atria squeeze. Okay, so that's what's happening with the P wave. All right. Then we stop at the AV node, and the signal very quickly rockets down the AV bundle to the bundle branches to the apex of the heart and up the Purkinje fibers. That's represented by the QRS complex. That's followed by ventricular systole, which happens right around here. And then the ventricles repolarize, getting ready for the next electrical signal for the next beat. And that's what gives us the T wave, okay? So depolarize, squeeze, repolarize, relax for the atria. Then depolarize, squeeze, repolarize, relax for the ventricles, all right? So that's what's happening in your typical EKG. If we interfere with this, then we interfere with the proper pumping of the heart. What will happen is we'll either have no squeezing or we'll have squeezing at an inappropriate time, okay? And the result is that the efficiency of the blood pump is going to be adversely affected. A lot of factors can act on the SA node to either increase or decrease the heart rate. Autonomic regulation we've already talked about, where the sympathetic and parasympathetic divisions of the autonomic nervous system affect the rate at which the SA node produces the impulses that lead to the heartbeat. The cardiac center in the medulla oblongata of the brainstem receives input from baroreceptors in the aortic arch and the carotid sinuses. A baroreceptor is simply a modified nerve cell that responds to changes in pressure by producing electrical impulses, in this case, which are sent back to the brainstem. It's also affected by emotions and impulses from the cerebrum and the hypothalamus, such as when we get angry or upset or frightened. Sympathetic neurons in the cardiac center will cause an increase in heart rate. The fibers for this exit the spinal cord through thoracic nerves to innervate the SA node, and they secrete norepinephrine, which is a chemical that's very similar to adrenaline. This causes the heart rate to increase, and it strengthens the force of myocardial contraction. Okay? So this is why I like to call the sympathetic division of the autonomic nervous system your freak out division. Okay? Think about what happens when you freak out, and you'll have a pretty good handle on the effects of the sympathetic division of the ANS. It's also stimulated by physical and emotional stress. The parasympathetic neurons in the cardiac center decrease the heart rate. These fibers exit via the vagus nerve, that's the 10th cranial nerve, and they innervate the SA and AV nodes, and their neurotransmitter is acetylcholine, which slows the heart rate. It's also stimulated by excessive blood pressure and emotional factors such as grief and depression. So, these responses should all make pretty good sense, okay? At rest, parasympathetic impulses are going to predominate. Increasing cellular needs are going to require sympathetic impulses. So the bottom line, if your blood pressure is dropping, the sympathetic division of the ANS will stimulate the SA node, and the heart will beat more rapidly and more powerfully. Okay? If your blood pressure is beginning to rise above levels that are good for the body, the parasympathetic division will kick in, and the rate and the force of heart contraction will be reduced. What you're looking at here is just a diagram showing you the wiring between the sympathetic division of the ANS and the electrical conduction system of the heart. There's the SA and the AV node, the AV bundle, the bundle branches, and the Purkinjes, and you can see where these fibers talk to those structures. And then the innervation that comes from the parasympathetic division. Remember, the vagus nerve is the major innervator here, and it's going to talk to the SA and AV nodes primarily. Okay? So, how do we understand what's happening with regard to our blood pressure? That's where the aortic and the carotid baroreceptors come in. Okay? An increase in pressure is going to cause them to fire more frequently. The result is that the cardiac center in the medulla oblongata will send out innervation via acetylcholine to the electrical conduction system of the heart to slow the rate and the force of contraction. Okay? By the same token, if the blood pressure monitored from the carotid and aortic baroreceptors is getting too low, then we're going to stimulate now the sympathetic division of the ANS, the same conducting centers in the heart, the SA node, the AV node, also the Purkinjes, okay, and the bundle branches. Other factors that affect the heart rate include age. The resting rate drops the older we get, and sex. Females have a slightly faster heart rate than males do, and of course physical condition. The better physical condition you're in, the lower and slower your heart rate is going to be. Temperature is also going to play a major role in regulating the heart rate. An increase in temperature usually increases the heart rate, while epinephrine will increase and strengthen the heart rate. Thyroxine, which is a hormone produced by the thyroid gland, which lies right in front of your trachea, produces a lasting increase in heart rate because of its effect on metabolic. Okay, what about the effect of ion levels in the blood? Well, calcium and potassium are two that we have to consider. Blood calcium levels are critical. Low levels will tend to slow the heart rate, while high levels tend to increase the heart rate and prolong the force of contraction. And for blood potassium, increased levels decrease both the heart rate and the force of contraction, while low levels can cause abnormal heart rhythms. This is one of the reasons why it's critically important for the body to maintain the levels of these ions within homeostatic limits. If they get too high or too low, you're going to adversely affect heart rhythm, and this could have a major impact on your health. Let's talk for a bit now about blood vessels. These are essentially a closed system of tubes that carry blood from the heart to body cells, and then from the body cells and tissues back to the heart. Arteries and veins have the same three basic layers. The tunica externa Tunica, by the way, is Latin for jacket. Okay, so this is the outer jacket of the blood vessel. It is basically fibrous connective tissue that provides elasticity and support. The tunica media is made up primarily of smooth muscle fibers that can cause a change in blood vessel diameter and, as a result, blood pressure. And the tunica interna is an endothelial lining of the blood vessel that has the ability to repel the formed elements so that they don't snag and rupture and leak. They also form a nice seal between the blood that they carry and the tissues surrounding them, so that really the only exchange that goes on happens when we get to the level of the capillaries. And we'll see that the capillaries have a fundamentally different arrangement of tissues that permit gas exchange and exchange of fuel and waste products than these other vessels do. So the walls of the arteries have the same basic structure. There's a few differences. Arteries have more smooth muscle and elastic connective tissue, while veins possess valves that prevent backflow. And the reason for this is that the pressure of the blood in veins tends to be dramatically lower than that in arteries. And in order to get the blood to return to the heart, we need these valves to prevent backflow working along with the smooth muscle tissue in the tunica media, but also with the skeletal muscle that surrounds those blood vessels to push the blood back up to the heart and enhance venous return. So if you guys don't move, if you're not active, sometimes what can happen is that the blood can pool, particularly in the lower extremities. You won't get adequate venous return to the heart, and that can have serious consequences. This is why the more active you are, the more venous return you have, the more efficient your circulation is going to be. So it's always suggested that you remain at least moderately active throughout the day. Another thing to realize, of course, about blood is that when blood flow slows or when blood pools, it tends to clot, and that can form emboli that can lodge in capillary beds and cut off blood flow to downstream tissues, causing them to infarct and die. Arteries always carry blood away from the heart, and they branch into smaller vessels called arterioles. The smallest vessels consist of just endothelial tissue and a few muscle fibers, and this is important in controlling blood flow and blood pressure. Capillaries are the most numerous and the smallest vessels. Red blood cells are forced to pass through these tubes one at a time due to their narrow diameter. The walls of the endothelia are thin enough to allow the exchange of materials between blood and cells. It's most abundant in active tissues. So anywhere that you see metabolically active tissue, you're going to see abundant capillary beds. They tend to be less abundant in certain connective tissues, such as ligaments and tendons, for instance. You can see here how we regulate blood flow to different tissue locations. We can do this through smooth muscle activity. Precapillary sphincters are kind of like bypass gates that regulate how much blood gets into a particular capillary bed. When the sphincters contract, you can see them situated here throughout this capillary bed. You're going to inhibit blood flow into the capillary bed, and the blood will simply bypass. It'll go straight through the capillary bed into the venous return and back to the heart. But when the sphincters relax, what happens is that blood flow through the capillary bed is permitted, and we're able then to deliver oxygen and nutrients while at the same time removing waste products. This is an intermittent activity in the body. At any one time, about 3/4 of the capillary beds are closed, but what happens is that the body's able to regulate which beds are open and closed from moment to moment. So we alternate beds that are closed off versus beds that are open in order to, in part, maintain our blood pressure and also to allow the tissues adequate perfusion so that they can get the nutrients and oxygen they need while allowing the removal of waste products. So essentially, it's going to be according to need. Um, the tissues are going to receive blood depending on things like the buildup of waste products, changes in pH. Okay, um, those are all factors that tend to promote um, dilation of these sphincters. If waste products build up or if pH drops too much, we tend to open these precapillary sphincters up and promote blood flow into the tissues. Same thing is true of oxygen levels. If oxygen levels start to get too low in tissue beds, the sphincters will open up and the beds will perfuse. Um, and the reverse is also true. If the tissues are adequately oxygenated and the concentration of waste products is low, then these beds tend to not perfuse. Okay, but again, this is a moment-to-moment thing. Materials pass through the interstitial fluid as they move between the blood and the cells. Remember, that's the fluid between the cells. Oxygen and nutrients diffuse from the blood into the interstitial fluid and into the cells, driven primarily by differences in concentration gradients, as all diffusion is. Remember that the blood that's coming to the tissues from the heart is oxygen rich and nutrient rich, whereas the situation around metabolically active tissue is that there tends to be low oxygen concentration compared to the blood and low nutrient concentration. And so diffusion is what drives a lot of this. Carbon dioxide and wastes go in the opposite direction because their concentration gradients are reversed. So as waste products that accumulate in the tissues build up, they're going to diffuse into the blood that's coming to the tissues from the heart and be carried away by blood flow. Water is also exchanged by osmosis, and that's going to be dictated primarily by the hydrostatic and osmotic factors that act in the tissue fluid and in the blood plasma. Okay, so what we have is what we call net filtration pressure, which is a combination of the effects of osmosis and hydrostatic pressure forcing water into and out of the tissues. Okay, and those factors usually stay relatively constant, but they can change in certain circumstances. Okay, blood flows from capillaries into venules, which unite to form veins, which form larger veins. The valves in large veins prevent blood flowing in the opposite direction back towards the tissues and aid venous return. The veins hold about 60% of the blood volume at any one time. The sinusoids of the liver and the spleen serve also as blood reservoirs. A sinusoid is simply a large, irregularly shaped, blood-filled area inside an organ or a tissue. Reducing venous volume can compensate for blood loss or increasing in muscle activity. Blood flows always from high to low pressure, as any fluid does. It's greatest in the ventricles and it's lowest in the atria. The ventricles create pressure when they squeeze, when they go into systole. The pressure decreases with increased distance from the heart. You can see here in this diagram that the heart would be located here. You could see the systolic pressure in the top trace, the diastolic in the bottom trace, and as we move from the aorta, right, the largest blood vessel in the largest artery in the body, into the large arteries and further from the heart, the total pressure begins to drop, as does the difference in pressure. And the reason for this is the resistance that's being experienced by the fluid as it flows through these vessels, and also the increasing distance from the pump that this fluid is experiencing. By the time we get to the arterioles and the capillaries, the pressures are so negligible that they're very difficult to detect. In the venules and the small veins and the large veins, you can see that it's practically zero. So how is it that we get blood back to the heart from the large veins, from the vena cava, small veins, etc.? Well, we rely on the action of the one-way valves, but also something called the skeletal muscle pump. In the skeletal muscle pump, what happens is as we move, the squeezing of the skeletal muscle on these blood vessels, on these veins, forces the blood through these valves and against gravity back up towards the heart. So the more active you are, the greater the venous return is going to be. Now, another factor is the thoracic pump. The fact that you breathe milks the inferior vena cava, and that also helps to pull blood up towards the heart through those one-way valves. If we didn't have these valves, we'd have a tough time maintaining venous return. Now, something else that we have to appreciate is that over a lifetime of use, sometimes these valves will weaken and collapse, and when they do, that produces a condition known as varicose veins. You've probably seen varicose veins. What happens as these valves weaken and collapse in these veins is they tend to bulge, and you tend to get venous stasis in this area. So varicose veins are not only uncomfortable, they're also a bit of a health hazard because blood flow through those areas slows to the point where it can produce clots, which can be thrown to places like the heart and the brain. Okay, so that's always a danger. Okay, respiratory movements, also critical for moving blood back towards the heart. The contraction of the diaphragm during inspiration drops thoracic pressure and increases abdominal pressure. The high pressure in the abdominal veins forces the blood into the thoracic veins and back up towards the heart. The velocity is going to decrease with an increasing number of smaller arteries and capillaries. So basically, the greater the cross-sectional area, the lower the flow is going to be. The velocity increases as blood flows back towards the heart into decreasing numbers of vessels. Flow is fastest in the aorta, and it's going to be slowest in the capillaries, and that has to do again with distance from the blood pump and with the resistance that the fluid experiences as it flows through those tubes. This is again due to the rapid circulation in the aorta, and in capillaries the slow movement of blood is critical so that we can exchange oxygen for carbon dioxide and metabolic waste products for fuel. Okay, so it's perfectly suited to its job, right? We want the aorta to really rocket that blood out of there. We want the capillaries to provide the tissues with the needed oxygen and nutrients and to remove those waste products so you don't create toxic conditions around the cells that cause them to die. Okay, blood pressure is simply a measurement of the force that the blood exerts on the walls of the vessels it's contained in. Arterial blood pressure in the systemic circuit is measured using systolic blood pressure, which is highest when the ventricles are squeezing, so during ventricular systole, and diastolic blood pressure, which is lowest during ventricular diastole. The pulse pressure is defined as the difference between the systolic and the diastolic blood pressures. It causes the pulse, which is the expansion and the contraction of the arterial walls. Now, this is important because if your pulse pressure is too high, then what you're doing is you're putting an excessive amount of force on the blood vessels, okay? And this is bad because especially in the capillaries, it could lead to rupture, and that will result then in a cutoff of blood flow to downstream tissues. What kind of things exacerbate or elevate the pulse pressure above what is advisable? Primarily problems maintaining your body condition. Okay, individuals, for instance, who are obese and inactive, what happens is that blood vessels tend to get longer instead of becoming more numerous, and they also tend to accumulate deposits that also further increase the pressure against those walls. And the result is excessive strain on the capillaries and the potential for those capillaries to rupture. Not good. With the changes that take place with arteriosclerosis, right, the hardening of the arteries that comes from deposits lining the walls, something else we have to consider is the fact that within those arteriosclerotic vessels, the pressure is high, but the flow is often restricted, and the result of that is also increased incidence forming emboli, forming clots. So that's something else we want to be aware of as well. Okay, what we're looking at here is just some of the major systemic arteries of the body. They're named for their location. We can start in the cephalic region up towards the head. You can see the temporal artery by the temples. The facial artery is going to run near the mandible and the maxilla. Okay, common carotid is in the neck. Axillary and brachial artery in the underarm and the upper arm region. Radial artery, femoral artery, popliteal artery, dorsalis pedis, and posterior tibial artery. Some of the major blood vessels in the body. Now, why do we point these out? These are also pulse points. Okay, a pulse point is characterized by an artery with very little overlying tissue. The result is very easy to palpate pressure changes in those vessels, and that can give us an indication of the overall health of the circulatory system. So what are some factors that affect blood pressure? Well, cardiac output, the volume of blood pumped by the heart in a single minute, which is determined by the heart rate and the blood volume pumped during a contraction. An increase in cardiac output is going to cause the blood pressure to climb, and a decrease is going to cause the blood pressure to drop. Blood volume is also going to have a role. An increase in blood volume will cause blood pressure to rise, while a decrease in blood volume will cause the blood pressure to drop because there's less fluid, in that case, pushing on the vessel walls. Peripheral resistance is a major factor in the regulation of blood pressure. This is the friction of blood against the vessel walls. The constriction of arterioles increases resistance and blood pressure, while the dilation of arterioles decreases resistance and blood pressure. Viscosity is also a major player. This is simply defined as resistance to blood flow. It's determined by blood cell concentration and plasma protein concentration. Basically, the thicker the blood is, the more stuff it's got dissolved in it, the greater the blood pressure is going to be, while a decrease in viscosity will tend to decrease blood pressure. So, as an example, conditions such as polycythemia will also cause an increase in blood pressure. Remember, polycythemia is an increase in the number of red blood cells in the plasma. Okay, peripheral resistance is controlled by the vasomotor center in the medulla oblongata, which increases the frequency of sympathetic impulses causing vasoconstriction. This increases blood pressure and velocity and accelerates oxygen and carbon dioxide transport. It also decreases a decrease in the frequency of sympathetic impulses is going to have the reverse effect. Okay, so here what we're looking at in terms of vasomotion is not so much parasympathetic, sympathetic dual innervation, but we're looking at changes in the frequency of innervation by the sympathetic division. So if we reduce those impulses, vasodilation will lower blood pressure and velocity. The activity of the vasomotor area is modified by epinephrine and impulses from higher brain centers such as the cerebral cortex, as well as impulses that come from pressure and chemoreceptors. Remember, pressure receptors are baroreceptors. Chemoreceptors are designed primarily to detect changes in carbon dioxide levels in the blood and also changes in the blood's pH. A decrease in blood pressure, pH, or oxygen tends to promote vasoconstriction. Okay, and so the idea here is that the vasoconstriction is going to up the amount of blood pressure. That's going to up the perfusion of the affected tissue, and that's going to accelerate exchange. Right? That'll remove the waste products. That'll more quickly deliver the oxygen, and the result will be a return to homeostasis. Blood vessels are also affected by local changes in blood composition, primarily oxygen and carbon dioxide levels, as well as pH. The effects can override vasomotor control, and this can increase the rate of exchange of materials between the cells and the capillaries that perfuse them. As an example, a decrease in oxygen and an increase in carbon dioxide will cause vasodilation to increase blood flow to a particular tissue. And this is important because if we don't do this, we're not going to get the oxygen and fuel we need to make ATP to allow the tissues to remain viable. At the same time, we're not going to remove the waste products quickly enough to not create toxic conditions that could end up killing or severely impairing cellular function. Okay, so let's talk a little bit about some of these circulatory pathways in the body. The pulmonary circuit is made up of the pulmonary trunk and arteries which carry deoxygenated blood to the lungs. The oxygen diffuses from the air into the blood. The CO2 goes in the opposite direction, and then the pulmonary veins carry the oxygenated blood from the lungs to the left atrium. And remember that the veins are going to carry blood that's going to be bright red, right, because of its high oxygen content. So here we have veins with bright red blood, arteries with blood that's darker red, which appears blue through the walls of the vessel. Okay, that's opposite in the systemic circulation, where the arteries carry bright red blood and the veins carry blood that's dark red and as a result has a low oxygen content. Okay, systemic circuit carries oxygenated blood from the heart to the body cells and returns deoxygenated blood from these cells to the heart. The left ventricle pumps the blood into the aorta, which branches and branches and branches, eventually leading into capillary beds. Carbon dioxide diffuses out of the tissue cells and into the capillaries, again guided by its concentration gradient. Blood leaves the capillaries and enters the venules, eventually into the larger veins, eventually into the vena cava. The superior vena cava drains areas above the heart back to the heart, and the inferior vena cava drains areas from below the heart back to the heart. So what are some of the major branches of the aorta? Well, here's a diagram showing you some of the major blood vessels, again named for their location. The coronary arteries supply oxygen to the heart muscle itself, while the brachiocephalic artery and the left common carotid bring blood up to the brain. Okay? The left subclavian brings blood from below the collarbone and towards the brachial region. The left subclavian eventually becomes the axillary and then the brachial artery, okay, which then eventually are going to branch into the radial and ulnar arteries and then into the palmar arches and the digital arteries. The intercostal arteries run between the rib muscles and the thoracic walls. The celiac artery is basically right below the diaphragm. Okay, so the diaphragm, let's draw the diaphragm in here so you can remember where it is. We'll do it in black. The diaphragm runs right through here. Okay, so that's the diaphragm. We'll write it down here, diaphragm. Okay, and there's three branches of the celiac artery which branches right under the diaphragm, and they supply the organs that lie in those regions. They are the gastric artery, which hit the stomach, the splenic artery, which goes to the spleen and pancreas, and the hepatic artery, which of course goes to the liver. The superior mesenteric artery goes to the first part of the large intestine, while the renal arteries hit the kidneys, and the gonadal arteries hit the testes in males and the ovaries in females. The lumbar arteries hit the walls of the abdomen and in the back. Okay, so these are named primarily for the tissues that they serve. Okay, that's a good way to go about learning these. The inferior mesenteric artery hits the distal half of the large intestine. The common iliac arteries are named for their proximity to the ilium of the hip. They hit the lower portions of the trunk and the legs. Arteries that run up to the head and the neck include the brachiocephalic artery, which forms the right common carotid and right subclavian. The left common carotid artery and left subclavian are independent branches coming off the aorta, and you can see them here and here. Okay. The common carotid artery forms the external and carotid artery, which is the neck, the face, and the scalp, and the internal, which supplies the brain. The subclavian arteries form the vertebral arteries, which hit the neck and brain. You can see them from this lateral view here. Okay, so notice again, subclavian artery, that's going to head to the axillary artery and then come the brachial artery, and then up here, right, you can see the common carotid going into the external and internal carotid. There's the internal, there's the external carotid. You can feel the external carotid when you take your pulse in your neck. Okay, and then you can see it branching the internal carotid deep into the brain, where it hooks up with the cerebral arterial circle, or also known as the circle of Willis, while the external carotid branches into arteries that supply tissues that overlie the lobes of the brain, such as the temporal, the frontal, the parietal, occipital, and so forth. Okay, in the shoulders and the arms, we have the subclavian artery forming the axillary artery, which hits the thoracic wall and the axillary region, the brachial artery through the upper arm, and then the radial and ulnar arteries, which run next to those bones on the finger, on the little finger, and on the thumb side of the forearm. And then once we're in the hands, we form the palmar arches and the digital arteries. In the lower extremities, the right and left common iliac branch off the abdominal aorta to form the external and internal iliac arteries. The internal iliac artery hits the pelvic wall, pelvic organs, external genitalia, and medial thigh, while the external iliac hits the anterior pelvic wall and continues as the femoral artery down into the upper leg, hitting the anterior and medial thigh muscles. The deep femoral artery hits the posterior and lateral thigh muscles, while the popliteal runs behind the knee and hits muscles of the thigh and leg. The anterior tibial artery runs in front of the tibia and hits the anterior and lateral leg, while the dorsalis pedis artery runs across the top of the foot and supplies the ankle and the foot. Posterior tibial runs behind the tibia and hits the posterior leg, ankle, and plantar, that should be a T there, plantar foot surface. That's the sole of your foot. The peroneal artery hits the lateral leg muscles. Sometimes you call this the fibular artery, which I prefer since it more accurately describes the location. The systemic veins run in the opposite direction, but they have many of the same names. They merge to form the superior and inferior vena cava, and this eventually empties into the right atrium. So veins that drain the head and the neck include the external jugulars, which drain the superficial head and neck, and the subclavians, which drain the external jugular veins and the vertebrals. The vertebral veins drain the brain and the neck, while the internal jugulars drain the brain, face, and neck. The brachiocephalic draining the internal jugular vein and subclavian vein, and then into the superior vena cava, which dumps back into the heart. The superior vena cava drains the right and left brachiocephalic veins. The veins that drain the shoulders and the arms include the radial and ulnar veins that drain the deep forearm, the brachial vein, which drains the deep arm and the radial and ulnar veins, the cephalic vein, which drains the lateral superficial hand, forearm, and arm, and the basilic vein, which drains the medial superficial hand, forearm, and arm, the axillary, which drains the basilic and the brachial veins, and the subclavian, which drain the axillary and cephalic veins. The anterior and posterior tibial veins drain the foot and the deep leg. The popliteal drains the anterior and posterior tibial and small saphenous vein. The small saphenous vein drains the superficial posterior leg, while the peroneal drains the lateral leg, and the femoral drains the deep thigh, the hip, and the popliteal vein and the peroneal vein. The great saphenous drains the medial and superficial foot, leg, and thigh. The external iliac draining the great saphenous and femoral. The internal iliac draining both the external and internal iliacs, which drain the upper thigh and pelvic area, while the common iliac drain the external and internal iliacs. And then finally, the inferior vena cava, draining both common iliacs, brings the blood back up to the heart. We look at the abdominal and thoracic wall. The azygous vein drains the thoracic and abdominal walls, the intercostal veins, and the ascending lumbar vein from the lower back. The ascending lumbar vein drains the wall of the abdomen. The inferior and superior mesenteric veins drain the large and small intestine, while the gastric drains the stomach, the splenic drains the spleen, the pancreatic draining the pancreas, and the hepatic portal vein is a special vein draining blood from the small intestine and the spleen and the stomach, and it goes into the back of the liver through an area called the portia hepatis. The pancreatic vein also merges with it. And what happens inside the liver is that the nutrients in the blood are stored temporarily as fat or as glycogen, or are passed on to the rest of the body if there's an immediate need. At the same time, the blood that goes in through the portia hepatis is also checked for the presence of pathogens. So the liver is a pretty important organ. It acts as a blood cleaning center. It also is important, acting as your body's kitchen and pantry. Okay, all of these veins form the hepatic portal system that lets the liver monitor and adjust the composition of blood coming from the GI. The blood flows to the liver where materials are removed or added. The blood then enters the hepatic veins, which empty into the inferior vena cava, which heads back up to the heart. The renal veins drain the kidneys, while the gonadal veins drain the ovaries in females and the testes in males. The renal and gonadal veins drain into the inferior vena cava. Okay, let's finish up talking about heart disorders. They can be grouped into heart disorders and blood vessel disorders. Okay, let's talk about some disorders of the heart and the blood vessels. We'll group them into heart and blood vessel disorders. Our first disorder is a heart disorder. It's called arrhythmia. This is an abnormal heartbeat caused by factors such as damage to the conduction system of the heart. Remember, that's the SA-AV node, the AV bundle and bundle branches and Purkinje fibers. Drugs can also cause arrhythmias. Examples of arrhythmias include bradycardia, which is a slowed heart rate, less than 60 beats per minute, tachycardia, which is over 100 beats per minute, and heart flutter, which is a heart rate between 200 and 300 beats per minute. Fibrillation is very rapid vibration of the heart chambers with uncoordinated contraction, and this results in no blood pumping. Fibrillation comes in two forms: AFib, which is a fluttering of the upper chambers, which does not present an immediate health hazard, and VFib, which is fibrillation of the lower chambers, which is a life-threatening emergency. We generally deal with this using an AED, okay, in an emergency situation. You've probably seen those in places where you work. They're embedded in the walls. AED stands for Automated Electronic Defibrillator, and you don't need to have any fear related to using these, because the AED, as you'll see in this week's activity, actually tells you what to do and when to do it. Okay, it'll talk to you through the entire procedure, telling you exactly how to use the device. So don't worry if you don't have previous training. Congestive heart failure is an inability of the heart to pump the blood that's returned to it. We see fatigue, edema, and accumulation of blood in the organs. One contributing cause is arteriosclerosis. This is hardening of the arteries, resulting in an increase in blood pressure, and as a result, an increase in the amount of work the heart has to do. Heart murmurs are unusual heart sounds often associated with defective valves, while MI is a fancy word for heart attack. This is the results in the death of the myocardium due to coronary artery blockage, and it can be an immediate life-threatening emergency. Pericarditis is an inflammation of the pericardium from viral or bacterial infection. Aneurysm is a weakened blood vessel that bulges and can potentially rupture. Okay, this can lead to a fatality if it occurs in the heart or in the brain. Arteriosclerosis is hardening of the arteries from calcium deposits that accumulate in the tunica media, causing blood pressure to increase and increasing the risk of emboli, and as a result, heart attack and stroke. Atherosclerosis is the formation of fatty deposits in the tunica interna of the arteries. These plaques reduce the lumen size and increase the probability of blood clot formation. By the way, that should be plaque with a Q, not a G. Hypertension is chronic high blood pressure over 140/90. Prehypertension is a systolic pressure between 120 to 139, and a diastolic between 80 and 89. Causes include stress, smoking, and being out of shape. Phlebitis is inflammation of a vein. It's most common in the legs. Thrombophlebitis is the formation of blood clots at a site of inflammation. Varicose veins are weakened valves that result in the bulging of blood vessels in the area of the valve malfunction. This causes venous stasis and can lead to emboli. Causes include heredity, pregnancy, obesity, and what we call a sedentary lifestyle. Hemorrhoids are a particular type of varicose vein that occur in the anal canal. Hemorrhoids are often joked about, but they're no laughing matter because of their location. If they bleed, they represent a significant infection risk, which could lead to sepsis, which could be, again, a medical emergency. So if you suspect that you have hemorrhoidal tissue, make sure and see your GP or your proctologist, and he will check you out. Okay, stay tuned for a quick review of the functioning of the heart, and also make sure to look over this week's activities that deal with blood vessels, the action of the heart, and the structure and function of blood. And I'll see everybody in the next podcast. Thanks for listening.
The heart is a hollow muscular organ, cone-shaped and roughly the size of a fist. It is located in the chest, behind the sternum, and between the lungs. The heart sits slightly off-center. The heart consists of 4 hollow chambers. The 2 upper chambers are called atria, and the 2 lower chambers are the ventricles. The right atrium receives blood from distal parts of the body by way of the superior or inferior vena cava. To keep all the blood flowing in the same direction, the heart has a system of one-way valves. All of the valves consist of leaflets that open and close in response to changing pressures within the chambers. Then blood passes through the tricuspid valve to the right ventricle. The right ventricle pumps the blood out of the pulmonic valve to the pulmonary artery and on to the lungs for a new supply of oxygen. Oxygenated blood returns via the pulmonary vein to the left atrium, and from the left atrium it passes through the mitral valve and into the left ventricle. When the left ventricle contracts, the oxygenated blood is ejected through the aortic valve to the aorta and distal body tissues. The walls of the left ventricle are 3 times thicker than those of the right side because it takes more pressure to reach distal parts of the body. The septum effectively divides the heart into right and left sides to form 2 separate pumps. The 2 pumps of the right and left heart function in close syncopation. This is a closed system, so the blood continues to pump and flow in a continuous cycle.
Action potentials originate in the sinoatrial node and travel across the wall of the atrium from the sinoatrial node to the atrioventricular node. Action potentials pass slowly through the atrioventricular node to give the atria time to contract. They then pass rapidly along the atrioventricular bundle, which extends from the atrioventricular node through the fibrous skeleton into the interventricular septum. The atrioventricular bundle divides into right and left bundle branches, and action potentials descend rapidly to the apex of each ventricle along the bundle branches. Action potentials are carried by the Purkinje fibers from the bundle branches to the ventricular walls. The rapid conduction from the atrioventricular bundle to the ends of the Purkinje fibers allows the ventricular muscle cells to contract in unison, providing a strong contraction. Ventricular contraction causes the atrioventricular valves to close, which signals the beginning of ventricular systole. The semilunar valves were closed during the previous diastole and remain closed during this period. Continued ventricular contraction increases pressure in the ventricles above the pressure in the aorta and pulmonary trunk, causing the semilunar valves to open. When the ventricles relax and their pressures drop, blood flowing back toward the relaxed ventricles causes the semilunar valves to close, which is the beginning of ventricular diastole. Note that the atrioventricular valves remain closed. When the pressure in the ventricles becomes lower than the pressure in the atrioventricular valves open and blood flows into the relaxed ventricles. This accounts for most of the ventricular filling. The atria then contract and complete the ventricular filling.